แล้วการบริการทางสื่อสารนี่จะดีนะฮะซึ่งตัวนี้เนี่ยยังต้อง
โลกวันนี้ใช่ไหมคะยังไงหนุ่ยรบกวนช่วยกรอกใบอันนี้นิดนึงนะคะเผื่อได้ไหม
พระราชทานกราบบังคมทูลทราบฝ่าละอองพระบาทข้าพระพุทธเจ้านายทัพชัยสุมิตรอธิการบดีจุฬาลงกรณ์มหาวิทยาลัยรู้สึกสำนึกในพระมหากรุณาธิคุณที่ได้ฝ่าละอองพระบาททรงพระเมตตาเสด็จพระราชดำเนินมาทรงฟังปาฐกถาในรายการสารสัมพันธ์สู่สันติวัฒนธรรมที่จุฬาลงกรณ์มหาวิทยาลัยร่วมกับมูลนิธิสันติภาพนานาชาติและธนาคารกสิกรไทยจำกัดมหาชนจัดขึ้นเมื่อวันพุธที่ 19 พฤศจิกายน พุทธศักราช 2546 และทรงพระมหากรุณาเสด็จพระราชดำเนินมาทรงฟังปาฐกถาในรายการดังกล่าวอีกวาระหนึ่งในวันนี้พระมหากรุณาธิคุณล้นเกล้าล้นกระหม่อมหาที่สุดมิได้ การแสดงปาฐกถาวันนี้เป็นครั้งที่ 6 ในรายการสารสัมพันธ์สู่สันติวัฒนธรรมที่จุฬาลงกรณ์มหาวิทยาลัยร่วมจัดขึ้นเพื่อเฉลิมพระเกียรติพระบาทสมเด็จพระจุลจอมเกล้าเจ้าอยู่หัวในโอกาสที่วันพระบรมราชสมภพครบ 150 ปี ในหัวข้อการนำผลการวิจัยเกี่ยวกับไนเตรตออกไซด์มาประยุกต์ใช้เพื่อการพัฒนาเวชกรรมใหม่โดยศาสตราจารย์เฟรดมูรัดผู้ได้รับรางวัลโนเบลสาขาการแพทย์วันนี้ข้าพเจ้าขอให้ทานพระราชานุญาตเบิกอาจารย์วลัยกัญญาปราศัยทำหน้าที่ผู้ดำเนินรายการด้วยเกล้าด้วยกระหม่อม
ขอพระราชทานกราบบังคมทูลทราบฝ่าละอองพระบาทข้าพระพุทธเจ้านางสาววลัยกัญญาพลาไสยขอพระราชทานพระราชานุญาตดำเนินรายการเป็นภาษาอังกฤษควรไม่ควรแล้วแต่จะทรงพระกรุณาโปรดเกล้าโปรดกระหม่อม Your Royal Highness, distinguished guests, ladies and gentlemen, Professor Ferid Murad has always known what he wanted. At age 12, in an essay for his 8th grade, he wrote about the first 3 choices of his career: a doctor, a pharmacist, and a teacher. He ended up doing all of that. From the beginning of his career as a scientist, Professor Murad has put a lot of time and effort in researching nitric oxide and its biological and physiological effects in the human body, and some of his colleagues believed that he was crazy to have spent so much time in something that was such a far-fetched idea. But his perseverance, determination, and perhaps a stroke of luck paid off in the form of the Nobel Prize for Medicine in 1998. 21 years after his first paper about nitric oxide was published in 1977. The scenario has indeed changed since then. There were about at least 50,000 scientific papers published about nitric oxide. People also listened to him. But as he put it, I'm no more brilliant than I was before the announcement, but now everybody is listening. And indeed we all are. With that, Your Royal Highness, distinguished guests, ladies and gentlemen, please welcome Professor Ferid.
It's unusual and often rare for a scientist to see his research over his lifetime have a large influence on patient care. Of course, that's why we go into medical research, and to see that happen has been very, very exciting for me. I think that my research has influenced the health care of probably millions of people around the world, and I think in the next decade it will influence millions more. I was very fortunate as a student, a medical student and graduate student, to work in Cleveland with 2 mentors, Earl Sutherland and Ted Roll. I joined their laboratory a year after they discovered the first intracellular second messenger, cyclic AMP. It became apparent over that next 5 or 10 year period that cyclic AMP was responsible for mediating the effects of many, many hormones in the body. They began their work by looking at the glycogenolytic effects of adrenaline and glucagon, the steroidogenic effects of ACTH, and the effects of antidiuretic hormone on water handling in bladder and the kidney. And subsequently, many, many other hormones were found to increase the accumulation of this messenger molecule, cyclic adenosine monophosphate. The concept of cellular communication is, I think, a very old concept, probably first introduced more than 100 years ago by Pavlov. Pavlov was a Russian physician and physiologist in St. Petersburg, Russia, who had a patient with a gunshot wound to his abdomen. This patient developed a fistula, a channel, a communication from the stomach to the exterior. And whenever this patient would smell food or see food, he would increase his gastric secretions. This, of course, led, for those of you who have taken psychology courses, to what's called conditioning. He developed dogs with a fistula to the stomach, and every time he showed them food or let them smell food, their gastric secretions would increase, but he would also ring a bell. And after a period of time, he didn't have to show the dogs food anymore. He just rang the bell, and they increased their gastric secretions. This was the really first scientific demonstration that the brain and the sensory systems were talking to the stomach. They influence the physiologic function of the stomach by some communicative network. The concept of cell communication today is very widely accepted. Many, many cells in the body talk to other cells. They communicate. It's a very complicated network. It's almost like an internet system with thousands and thousands of emails going back and forth between one cell type and another. This concept is summarized for you in this slide. These are three different populations of cells. Let's call this a brain cell, a neuronal cell. Let's call this an endothelial cell, the cells that line the inside of blood vessels. And let's call this a smooth muscle cell in the blood vessel wall. This brain cell is going to talk to this cell by producing a substance that Earl Sutherland called a first messenger. We give these first messengers a lot of different names. We call them hormones, we call them neurotransmitters, we call them paracrine substances, we call them autocoids, we call them cytokines, we call them growth factors. There are hundreds and hundreds of these first messenger molecules. Some of these structures are very simple molecules, some are very complicated proteins and peptides. But the concept is the same. These substances, when they're released into the bloodstream, will seek out a target cell somewhere in the body to perturb it and alter its biology. And it does this by interacting with a protein, often in the membrane of this target cell, that we call a receptor. Because of the three-dimensional conformational fit of the first messenger molecule with this receptor, like a hand in a glove or a key in a lock, only the right molecules are identified by the correct appropriate receptors. That's where the specificity of this interaction takes place. It's like 1,000 emails going out, but only one computer terminal will pick up the appropriate email if it's got the right code password. This interaction of these molecules, this protein receptor with these first messengers, perturbs a biochemical pathway in these cells to permit the intracellular accumulation of what Earl Sutherland called intracellular second messengers. The first such intracellular second messenger was cyclic adenosine monophosphate, cyclic AMP, derived from ATP. For that work, he received the Nobel Prize in 1971. Today we know there are but 10 or 12 or 15 of these intracellular second messengers. Besides cyclic AMP, there is calcium, there is cyclic guanosine monophosphate, a cousin of cyclic AMP. There is nitric oxide, there is diacylglycerol, derived from lipids, phospholipids. There are some peptides, there's some other molecules. So there are hundreds of these, and a dozen or so of these intracellular messengers. These intracellular messengers do the biochemistry inside of the cell without the hormone having to get inside. The hormone only works at the surface by perturbing this biochemical cascade to make these messengers that do the business. What is unique about nitric oxide as an intracellular second messenger is that it's a free radical, it's a gas, it's a very reactive substance, but it's also uncharged at physiologic pH. Because it's uncharged, it's very lipid soluble, and it can go in and out of cells quite freely. Other messengers can't do that. The first messengers usually don't get in, the second messengers usually don't get out. So nitric oxide is a unique messenger molecule that can function inside of cells, but it also can be released to influence the biology of other cells. It can feed back to the cell and say, I don't have enough information or I have too much information. And in biology, feedback loops, positive and negative, are very important for maintenance of homeostasis. Many endocrine diseases lack the feedback system, and you have too much of a hormone effect or too little, and therefore some disease. One of the things that nitric oxide does that we found 26 years ago is that it increases cyclic GMP synthesis, a cousin of cyclic AMP, and we'll talk more about that, in the cell in which it's made, but also in neighboring cells, and this leads to relaxation of the smooth muscle. This is one of the fundamental important mechanisms for smooth muscle relaxation, to control the diameter of blood vessels and the relaxation of other smooth muscle in the body, in the airways of the lung, or in the gastrointestinal tract, to influence the motility of the bowel, etc. Now, nitric oxide is a very old molecule. From the work of astrophysicists and astronomers, it's believed that this Earth is probably between 4 and 5 billion years old. And about 3.5 billion years ago, oxygen appeared in our atmosphere for the first time. But there's evidence that there probably was life on this planet before there was oxygen. And I suspect that this life originated in the deep pits of the ocean, near fissures and magma coming up and all this heat and all these minerals and chemicals in a boiling pot, and there were organisms trying to survive. They were eating each other, they were colonizing, making more complex multicellular organisms, and they had to communicate with each other. How did they communicate? Probably with very simple molecules: calcium, phosphate, nitric oxide, hydrogen sulfide. So I think that nitric oxide probably participated in evolution of life. I can't prove that because I don't know how to design that experiment. I wish I could. Nitric oxide became popular about 50 or 60 years ago when it was apparent that any fossil fuel that possesses nitrogen in its structure, when it's combusted with oxygen, leads to the formation of nitrogen oxides. Nitrogen can exist in 5 different valence states, so there are a family of nitrogens complexed with oxygen. The most simple is the nitrogen monoxide, which has a single electron in its outer shell, and by definition is a free radical, making it very reactive. Molecules want to fulfill their outer shell with the appropriate number of paired electrons. So it will often suck an electron off of another molecule and oxidize it. So nitrogen oxide, the monoxide, comes out of smokestacks, cigarette smoke, automobile exhaust, and the environmentalists became concerned about nitrogen oxides as well as other gases being emitted from the combustion of fossil fuels, because they deplete the ozone layer in the outer atmosphere, which is our protection as an ultraviolet filter to prevent global warming. What I'm going to tell you today is that not only is it a pollutant and a toxic substance, it's also how some drugs work. Some very important drugs: nitroglycerin, nitroprusside. Nitroglycerin was first synthesized by Italian chemists in 1847. It was first used clinically for angina pectoris, heart pain with impending myocardial infarction, in 1870. And while it had been used for more than 100 years, none of us understood its molecular mechanism of action. Well, it turns out that nitroglycerin is a precursor of nitric oxide. When you take it, in the body, it's converted to nitric oxide. The same is true with another important cardiovascular drug, nitroprusside, a drug that we use in the hospital to maintain blood pressure in severely hypertensive patients or in patients that had a myocardial infarction. We want to lower the blood pressure to decrease the workload of the heart. We give them drugs such as nitroglycerin and nitroprusside. It's also an endogenous substance in the body produced by virtually all cells, with a few exceptions. It's very ubiquitous. It's a very important fundamental messenger molecule. We'll talk more about that. This is a partial list of some of the biological effects of nitric oxide. It relaxes smooth muscle, airway smooth muscle, gastrointestinal smooth muscle, vascular smooth muscle. It controls the blood flow and the amount of perfusion of any organ or tissue, and therefore the nutrients delivered to it and the oxygen delivered as well. It inhibits the aggregation of platelets, so it prevents blood clotting, which is beneficial. Works differently than aspirin and the prostaglandins, but just as effectively. It participates in lots of inflammatory diseases. If you make too much of the nitric oxide, there's evidence that you modify proteins in the heart, the kidney, in the intestine, and colon, with inflammatory bowel disease, or in the joints with arthritis. It is responsible for killing bacteria and parasites. It's one of the first lines of defense with an infection. But if you make too much of it, the blood pressure drops and patients develop what's called septic shock, with a mortality rate of about 70% in intensive periods. So patients with very severe trauma, very severe infections, are making a lot of nitric oxide, more than they need. So the whole key to what I'm going to be telling you today is that while almost all tissues produce it, it's very important that you produce the right amount at the right time, at the right place. If you produce too little, you have disease. If you produce too much, you have disease. So the story, obviously, is much more complicated than I'm going to try to summarize for you today, with all the biochemistry and the multiple isoforms and pathways, but I'm going to give you an overview of how some of this can be done and how it's become a very promising area for drug discovery and development. It plays a role in penile erection. There are nerves that secrete nitric oxide as their neurotransmitter. Some nerves secrete norepinephrine, some secrete peptides, some secrete glutamate, some secrete a variety of things, but there are a population of nerves in the body, in the intestinal tract, and in the blood vessels in the penis that secrete nitric oxide. Many patients with various diseases have what's called endothelial dysfunction, and the nerve terminals and the endothelial cells in the blood vessels don't make enough nitric oxide. These patients have impotence, erectile dysfunction. How do we take advantage of that? Well, with drugs like Viagra, which inhibits the degradation of cyclic GMP to enhance the effects of the residual endogenous nitric oxide that you have in those blood vessels. And this will be apparent. We'll talk more about this in a second. For the first 10 years or so of my training, I emphasized cyclic AMP. I was the person in Earl Sutherland's laboratory that showed that cyclic AMP was responsible for the effects of adrenaline on the heart. The effects of epinephrine and adrenaline because the heart to pump harder is because it makes more cyclic AMP. And that was blocked by beta blockers, which become very popular for hypertension and maintenance of arrhythmias and blood pressure, etc. In the mid-1960s, after the discovery of cyclic AMP, a couple of chemists gave radioactive inorganic phosphate, P32, to rats, collected the urine, and found in the urine two major organic phosphate compounds. One was cyclic AMP, the other was cyclic GMP. This was the first demonstration that cyclic GMP, guanosine monophosphate, cousin of cyclic AMP, was an endogenous substance in the body. Well, if it's there, presumably it's going to be important. So several laboratories began to look for the enzymes that were responsible for its synthesis This enzyme is called guanylyl cyclase, which converts GTP, similar in structure to ATP, to cyclic GMP and pyrophosphate. This reaction is very, very analogous to another enzyme, adenylyl cyclase, that converts ATP to cyclic AMP. In fact, the enzymes have homology. They probably were derived from some common gene based on their sequence analysis of the adenylate cyclases and the guanylyl cyclase. Another group of investigators were looking at enzymes that hydrolyzed cyclic AMP and cyclic GMP, and found that there was an isoform of phosphodiesterase that selectively hydrolyzed the phosphodiesterase bond of cyclic GMP to inactivate it and convert it to 5' guanosine monophosphate. It turns out there are a complicated family of adenyl cyclases. There's a large family of guanylyl cyclases to make the story much more complex. And there is a large family of phosphodiesterase. It's probably about 10 different isoforms derived from 10 different genes. The type 5 phosphodiesterase is found predominantly in the wall of the blood vessel. This is the enzyme that selectively hydrolyzes cyclic GMP in the blood vessel wall, including the blood vessels in the corpus cavernosum of the penis, and that's the enzyme form that is inhibited by sildenafil or Viagra. That's how it works. It's like nitric oxide pushing the synthesis of cyclic GMP and diesterase pulling the cyclic GMP out of the system. It's as though you have a bucket of water with a tap, which is nitric oxide, that puts water in the bucket. The more you open it with more nitric oxide, the more water in the bucket, the more cyclic GMP. But there's a hole in the bucket. So it's going out as fast as it goes in, and the phosphodiesterase is the hole. If you plug that hole with your finger, Viagra, the bucket fills up with water, more cyclic GMP. That's simple. And this is very common in biochemistry, that the steady-state concentrations of substances are a function of the rate of synthesis as well as the rate of removal and degradation. Other laboratories started looking for potential targets of these messengers, and it was known that there's a protein kinase that is selectively activated by cyclic AMP, and sure enough, there is a protein kinase selectively activated by cyclic GMP. In fact, it turns out there are about 150 different protein kinases. When these second messengers act with their appropriate protein kinases, they activate these enzymes. These enzymes transfer the terminal phosphate of ATP to a protein substrate, and the protein becomes phosphorylated. This is the basis of signal transduction. Various hormones and ligands influence the production of a messenger. The messenger regulates the activity of the kinase, which regulates the phosphorylation of the protein. And when the protein is phosphorylated, its shape, its conformation changes. If it happens to be an enzyme, its activity is increased or decreased. It's really that simple. Except that we've got hundreds of these enzymes to play with to add to this complexity. So this story was beginning to evolve as I was finishing training, and I became very intrigued with cyclic GMP as another possible second messenger. And I set out, as I joined the faculty in Virginia, to answer two questions. How do hormones, neurotransmitters, perhaps other substances, precisely regulate the enzyme guanylyl cyclase to make cyclic GMP? If we understood how these hormones interacted with their receptors and transmit that information to the enzyme guanylyl cyclase to make cyclic GMP, we should be able to come along with compounds and drugs to enhance that coupling mechanism or block it as therapeutic agents for various diseases. That was my rationale. So I wanted to understand this molecular mechanism so that I could intervene to enhance or block these pathways. The second question, if you have cyclo-GMP in tissues, it has to be doing something, but what is it doing? We had no idea what its functions were. Well, we began our studies with guanylyl cyclase, and in our earliest experiments, we found that all the tissues we looked at had multiple activities in the cytosolic fraction of tissue homogenates, as well as the particulate membrane fraction. And these activities behave differently from each other, suggesting that we had multiple forms of the enzyme. At the time, this was not the case with adenylyl cyclase, which was always membrane associated. If there were different forms of the enzyme in different compartments of the cell, perhaps they were regulated by different categories of hormones. Some hormones would regulate the soluble isoform or cytosolic form. Others might regulate the membrane form. That would then create different intracellular pools of cyclo-GMP that might have different functions. So I thought that the problem was going to be much more interesting and challenging than my previous experience working with cyclo-GMP. Ultimately, we proved that that was the case. We purified these enzymes to homogeneity. We cloned them. We understand their genes. We understand their promoters. We developed a lot of tools and reagents in the past 25 years or more, 30 years. But in the earliest experiments, when we thought we had multiple isoforms, we were working with very crude preparations and we couldn't be absolutely sure. Because you can get misleading data in crude systems when you're doing biochemistry characterization, because there are other pathways and enzymes that compete for the substrate or the product that you're measuring, and you can get artifacts. And we knew that purification and characterization and cloning is a long-term investment. In fact, it was. It took us many, many years to do all that. But back in the early 1970s, we took some shortcuts. We said, let's introduce in our incubations inhibitors of nucleotidases, phosphatases, phosphodiesterase, various enzymes that were competing either for GTP as the product or cyclo-GMP that we were measuring. So we developed cocktails of various compounds, pyrophosphate, fluoride, azide, hydroxylamine, sodium nitrite, a long list of goodies, methylxanthines, which we knew were diesterase inhibitors. And while hormones, a couple of hormones, would increase cyclo-GMP accumulation in intact tissue, they failed to activate in the broken cell. Somehow, when we homogenized the tissue, we disrupted the ability of the interaction of the hormone with its receptor to talk to and regulate guanylyl cyclase. We disrupted something. Wouldn't work in broken cells. And if our goal was to understand the molecular coupling, it was imperative that we have a hormone effect in broken cells in order to pull the components apart, purify them, and put them back together in a reconstitution experiment to verify that they were important players. No such luck. Hormones wouldn't work when we disrupted the tissue. But accidentally, in this cocktail of materials that we added to our incubations, we found a couple of compounds that could activate the enzyme. Sodium azide, which is a poison, a toxic substance. You often throw it in the solutions that you store in the refrigerator in a cold room to inhibit bacterial growth. Sodium nitrite and hydroxylamine. Those three substances activate guanylyl cyclase. And that was quite a treat and a surprise. We didn't expect that. Accidental. So much of science is accidental. When we found that, it turned out, when we characterized these effects, that the azide effect required oxygen in our incubations. It was only working with some tissue extracts and not everything that we tested. It had a time lag of several minutes before the rate of the reaction became maximal. There was a lag. And we could enhance the effect of azide by putting in some reducing agents, particularly thiols. These experiments suggested that these substances, azide, hydroxylamine, and sodium nitrite, were probably being converted to something else. And we were determined to find out what that something else was, because we wondered if we knew what that something else was, maybe that's what hormones were using to regulate the enzyme. That was our thought at the time. We did a very simple biochemical experiment. We took a tissue extract, liver, supernatant, where azide would activate the enzyme. We took a heart supernatant where azide had no effect, a cerebral cortex supernatant where azide had no effect, and we combined them. We mixed liver soup with heart soup. The azide effect was blocked. And that's because heart possessed an inhibitor of the azide activation. This inhibitor was heat labile, non-dialyzable, presumably a protein. We purified it, it turned out to be two proteins, hemoglobin and myoglobin. If we mixed liver with cerebral cortex supernatant, the azide effect was enhanced, it was potentiated. Why is that? Because the liver was converting azide to this unknown activator. Cerebral cortex couldn't do that, that's why it didn't respond. But when we converted it with the liver present, it could activate the enzyme both from liver and cerebral cortex to augment the response. This factor was also heat labile, non-dialyzable. We purified it and turned it into catalase. So we had a family of heme proteins, hemoglobin, myoglobin, that were inhibitors, catalase that was an activator of the azide effect. We could replace the catalase with some cytochromes, with the horseradish peroxidase, and other heme proteins. These were important clues as to what this substance might do. We took these substances, azide, hydroxylamine, and sodium nitrite, and added them to cell cultures, tissue culture cells, slices from liver and cerebellum and other tissues, and found that they could increase cyclic GMP levels in intact tissue. So they worked both in tissue extracts, homogenates, as well as intact tissue. It was a hormone surrogate, if you will. It would replace the hormones in intact tissues, but it also worked in broken cells. Fortunately, one of the tissues that we were working with at the time was tracheal smooth muscle. I had known from my previous work that cyclic AMP relaxed smooth muscle. It relaxed blood vessels, airway smooth muscle, gastrointestinal smooth muscle. And I thought that perhaps cyclic GMP might antagonize the effects of cyclic AMP. So we developed a tracheal smooth muscle preparation where we could correlate biochemistry with the physiology, contraction, and relaxation of those preparations. We put azide, hydroxylamine, sodium nitrite on these tracheal smooth muscles. They elevated the cyclic GMP levels. No surprise, because it didn't win in other tissues. The big surprise was that the tissue didn't contract, it relaxed. And over the course of months, it became apparent that the accumulation of cyclic GMP was very tightly coupled to relaxation of smooth muscle, with dose response curves, time courses, etc. And we had found, for the first time, a function for cyclic GMP, smooth muscle relaxation. Not only did it relax airway smooth muscle, but also gastrointestinal smooth muscle, vascular smooth muscle. When we found these substances relaxing smooth muscle, we said, what happens with other smooth muscle relaxants? What does nitroglycerin do? What does nitroprusside do? And sure enough, they too would activate guanine cyclase, increase cyclic GMP, and cause relaxation. So we had a list of compounds that we called nitro-vasodilators. They included hydroxylamine, azide, sodium nitrite, some hydrazines, nitroglycerin, nitroprusside, nitrosoureas, nitrosamines. Nitrosamines are cancer agents that you get from eating hot dogs, all the nitrate you worry about the possibility of cancer in fruits, food materials. All of these substances have nitrogen. Some have acidos, some have amines, some have nitros, some have nitrosos, but they're all converted to an intermediate. And why do we think that the intermediate is nitric oxide? Well, that's because hemoglobin and myoglobin were inhibitors of the activation of all these substances, and we knew from some old literature from the 1940s and 50s that nitric oxide had a very high affinity for the heme prosthetic group in hemoglobin and myoglobin. And we suspected that nitric oxide was the culprit and the common mediator, and sure enough, that was the case. Nitric oxide would activate guanylate cyclase, and we could block that with hemoglobin and myoglobin. If we oxidize the monoxide to the dioxide, which is a very toxic brown gas, with hydrogen peroxide, potassium ferricyanide, and other additions, we lost the effect. We were quite excited that a free radical could activate an enzyme. This was unprecedented. Free radicals are supposed to be toxic. They're bad. They use spin-turner, they destroy proteins, they destroy genes. They're never good things, was the dogma. And for the first time, we had a free radical that could activate an enzyme, and I thought this was pretty exciting. It was a novel chemistry. Well, our colleagues were not quite so impressed. They said, why isn't nitric oxide inhibiting an inhibitor of guanylate cyclase? If you inhibit an inhibitor, you get an activation. So we were compelled to purify the enzyme to homogeneity, and we did. It took us 6 or 9 months. And when we purified the enzyme to a single protein, we still had an effect of nitric oxide. In fact, the quantity of nitric oxide required to activate the enzyme kept decreasing and decreasing as we removed other contaminating proteins and transition metals and other materials that were scavenging or acting as sinks to suck up the nitric oxide. With purified enzyme, we could activate the enzyme with nanomolar concentrations of nitric oxide. The enzyme was exquisitely sensitive to the free radical, and we could activate the enzyme as much as 400-fold, a huge effect at low concentrations. Quite exciting. This is what the effects of the nitro-vasodilator look like on a blood vessel. This is a segment from a rat aorta that has been pre-contracted with norepinephrine to get a tone on the tissue. And then after 5 or 6 or 7 minutes, we add an donor, a nitro-vasodilator, to the any of them. This happens to be nitroprusside. And within 10 or 20 seconds, cyclic GMP levels increase in the tissue, peak in about a minute, return to basal levels, and this is followed by a relaxation of the tissue that persists for a long period of time. So the messenger precedes the physiologic effect, but at late times they're dissociated. The messenger's gone, but the effect persists. Why is that? Well, the messenger has a short half-life. Most intracellular cycle messengers do. But the activation of the kinase and the phosphorylation of the protein substrates that are responsible for the effect have a protracted half-life. And if you look at late times, there are a variety of smooth muscle proteins that are still phosphorylated, even though cyclic GMP is back to control levels. And this is very common in signaling systems. So we proposed in a couple of review articles in 1978 that we had figured out the mechanism of action of the nitro-vasodilators, namely nitroglycerin and others. They were prodrugs that were being converted to nitric oxide to activate guanylate cyclase to make cyclic GMP, and that's how the smooth muscle relaxed. And as a pharmacologist, when you find the effect of an exogenous substance or drug, you should think about the possibility of it reproducing what is happening endogenously. For example, the effects of opiates, morphine, heroin, led to the discovery of enkephalins. There are lots of examples of that in the pharmacological literature. So because nitric oxide is mediating the effects of drugs, I said perhaps the effects of hormones to increase cyclic GMP might be attributable to the formation of nitric oxide from some endogenous precursor. I proposed that nitric oxide was going to be a normal endogenous second messenger. Not only was it a free radical activated enzyme, but the free radical is important in the body, and this was real heresy. And it was not a popular area. It took a long time for people to begin to believe this, probably a good 7 or 8 years. I was discouraged, of course, that they wouldn't listen to this story and believe it. But at the same time, it gave us an opportunity to get an awful lot of work done and dominate the field. And for a long period of time, we controlled the whole field. There was no one close. And that was comforting for all my trainees who developed the long publications. But that happens. I guess the point is that often in research, when you find something that is so different than unique and against the dogma, it's almost viewed as heresy. And it's difficult for others to accept it. Most scientists, the majority, like to follow the leader and do what's been done. I don't view that as research. I think if you're not working on the edge of the envelope, it's not research. You've got to do something different than you might do. Robert Furchgott is a professor at the State University of New York in Brooklyn and a vascular biologist. And for many years, he contributed to our understanding of the effects of various hormones on blood vessel function. The effects of epinephrine and norepinephrine and other drugs with receptor characterization, etc. It had been known for a long, long time that some hormones and drugs, when given to animals or man, would dilate blood vessels and lower blood pressure. Acetylcholine, bradykinin, histamine, a variety of drugs. But when you added these substances to vascular segments in an organ bath in the laboratory, they did nothing. They never worked. Why was that? Well, it turns out that Furchgott found that when you make blood vessel segments in the laboratory, you often destroy the integrity of the endothelial lining of the blood vessel. Because you open up the blood vessel, you put it on your fingertip, you put it on a paper towel, you touch the endothelium, it's very sensitive, and you destroy it. If you're careful to maintain the integrity of the endothelium, these substances would cause relaxation. So he developed the concept of endothelial-dependent vasodilators. These vasodilators, a long list of them, would cause relaxation only if the endothelium were intact. And he found from a very clever experiment with a sandwich of blood vessels, he had one blood vessel strip with endothelium, another blood vessel strip with no endothelium. He put them together, and when he put on an endothelial-dependent vasodilator, both strips relaxed. And that's because something came out of the endothelium, and it went both ways to relax both split muscle segments. This substance he called endothelial-derived growth factor, EDRF, endothelial-derived relaxin factor. And it was very labile. It had a half-life of only 2 seconds. It was very reactive. He characterized it to some extent. He presented this work at a seminar that I heard in 1980. And after a seminar, I told him that this is probably going to be a free radical of some kind. It probably worked through cyclo-GMP. And sure enough, that was the case. And we went on to characterize that. This is a confirmation of Furchgott's experiment. Bob works with gravity segments, and we work with radioactive. But here again, we pre-contracted this radioactive with norepinephrine, and then at 0 time, we add one of these endothelial-dependent vasodilators, acetylcholine, histamine, bradykinin, ATP, thrombin, and a long list. And within 10 or 20 seconds, cyclo-GMP levels increase, return to basals, and this is followed by relaxation that persists for a long period of time. But this only occurs if the endothelium is intact. If you remove the endothelium, cyclo-GMP levels are not elevated, the tissue does not relax. Furthermore, the basal cyclic GMP levels decrease about 50%. The cyclic GMP-dependent protein kinase activity ratio also decreases, as does the phosphorylation of a variety of proteins as determined by p32 incorporation in proteins displayed in two-dimensional gels. Basal cyclic AMP levels also decrease, and that's because you're removing the source of prostacyclin that comes out of the endothelial cell that activate adenylate cyclase to make cyclic AMP. So the endothelium is talking to the underlying smooth muscle to control the synthesis of both cyclic AMP and cyclic GMP. And the substances that come out, prostaglandins, EDRF, this reactive substance, go in both directions. So not only do they relax smooth muscle, they also influence the formed elements in the bloodstream. They inhibit aggregation of platelets. They influence the polys, degranulation, other things. And that's very important in wound healing and inflammatory events and other things that we'll talk about. So by the early 1980s, we recognized that there were two pathways, two different categories of compounds and drugs that resulted in enhanced cyclic GMP formation. The nitro-vasodilators by producing nitric oxide, the endothelial-dependent vasodilators by producing EDRF, both of which activated the enzyme to make cyclic GMP. We then spent a few years showing that this resulted in the activation of cyclic GMP-dependent protein kinase in the tissue, which resulted in the phosphorylation of a variety of smooth muscle proteins, some of which we were able to identify, that then led to relaxation of the smooth muscle. This cartoon sort of pulls this story together for you. This is to represent a blood vessel with its endothelial lining on the left and the smooth muscle on the right. In red are three different categories of vasodilators that work by increasing cyclic GMP synthesis. The nitro-vasodilators either spontaneously liberate nitric oxide based on the local pH, oxygen tension, etc., or they're enzymatically converted to nitric oxide, as with, say, nitroglycerin. There's an enzyme that converts it to denitrates to make nitric oxide. The nitric oxide activates the soluble isoform of guanylate cyclase to convert GTP to cyclic GMP. How does it do it? Soluble guanylate cyclase is a heterodimer with an alpha-beta subunit. The beta subunit has a heme prosthetic group ligated to the histidine 105 position. Nitric oxide binds to the ferrous ion in the heme, pulls the porphyrin away from the beta subunit, and permits the activity of the enzyme to increase. Basically, that's how it works. Because you can mutate that histidine, get rid of the heme, and the enzyme is fully active in the absence of nitric oxide, which I think will be very important for gene therapy for some diseases down the road. The cyclic GMP activates the cyclic G-dependent protein kinase. This phosphorylates a variety of smooth muscle proteins. These phosphorylated proteins result in decreased concentrations of calcium in the cell. The lower calcium means decreased phosphorylation of myosin light chain. Myosin light chain kinase is calcium calmarginally dependent. So when the myosin light chain is dephosphorylated, you get relaxation. What happens is when you active myosin filaments is that when you phosphorylate myosin, it's still not full together to get contraction. You dephosphorylate the myosin, the filaments slide apart to get relaxation. The endothelial-dependent vasodilators do exactly the same thing, but they require the integrity of the endothelium. What they do is they elevate cytosolic calcium, which is required for an enzyme called nitric oxide synthase that converts arginine in our diet to EDRF, which is also nitric oxide. We learned in the mid-1980s from several laboratories that the EDRF molecule is in fact nitric oxide. So it obviously is going to work through the same pathway. A third category is called the atrial natriuretic factors or atriopeptins. In the early 1980s, de Bold and Canada discovered that there were granules in cardiac atria that produced a peptide hormone that was capable of dilating blood vessels and capable of influencing the kidney's handling of salt and water. So the heart is an endocrine gland. It has a hormone that regulates function of blood vessels and the kidney. And when we saw that work, we wondered if it worked through cyclic GMP. And sure enough, it does, but it activates the particulate isoform of guanylate cyclase. In fact, it turns out the particulate isoform of guanylate cyclase is one of the receptors for atriopeptins. So there are three categories of materials that work by enhancing the intracellular concentration of cyclic GMP in the smooth muscle. The nitro-vasodilators and atriopeptins are direct acting on the smooth muscle. You don't need the integrity of the endothelium. The endothelial-dependent vasodilators only work if the endothelium is normal. Why is that important? Well, there are lots of diseases that have endothelial dysfunction. Hypertension, diabetes, atherosclerosis, cigarette smoking. The endothelium might look normal, but biochemically it's abnormal, and it doesn't produce enough nitric oxide. How do you get around that? By giving a direct-acting vasodilator, a nitric oxide donor, or atriopeptins. That's why we treat patients with angina with the nitric dilators. We don't treat them with hormones that are endothelial dependent because they've got atherosclerotic disease in their cardiac vessels. Okay? And we'll talk more about that in a second. The enzymes that make nitric oxide from RG are called nitric oxide synthases, and there are three different forms of the enzyme. They've been given different names. They were initially called neuronal, inducible, and macrophage, and endothelial. And we suggested some years ago that we rename them NOS1, 2, and 3 in the chronological order in which they were first characterized, purified, and cloned. These genes for these enzymes reside on different chromosomes. Almost all cells have one or another of these enzymes. Some have all three of them, some have one, some have two. There's a lot of homology between them, but yet they differ. And there are compounds that inhibit one or another a little bit more selectively than other compounds. Interestingly, the NOS2, the inducible form, is normally not present in tissue. The gene is there, but there's no transcript and there's no protein, unless there's been an inflammatory insult. If you have inflammation, the pro-inflammatory cytokines, the factors that come from infections like endotoxin, induce the gene to make the transcript to make the protein. So if you see the protein, if you see the transcript, it's a sign of inflammation. So it's become an inflammatory marker. Why is this important? Well, it turns out in serious infections, you make a lot of nitric oxide by NOS2 to kill the bugs. It's the first line of defense for infection, even in plants. If you see a little node or a scar on the side of a plant, that's because it's made nitric oxide to kill that invading organism. The same thing happens if you damage your skin or anything in your body. But there are situations where you make too much of the NOS2, too much nitric oxide, and the blood pressure drops as in septic shock. Or inflammatory diseases, nephritis, colitis, Alzheimer's, arthritis, etc. Perhaps in tissue transplantation. You're turning on the NOS2 system, you make a lot of VNO, and VNO becomes toxic, and there's a problem. These enzymes are much more complex than I'm telling you. They've all been cloned. Fragments and even the whole enzyme has been studied in X-ray crystallography. They have a complicated array of cofactors. The co-substrate is oxygen and NADPH, but they also use flavines, tetrahydrobiopterin. They have a heme prosthetic group. All the proteins that interact with nitric oxide, that either make it or respond to it, have a heme prosthetic group. And it's because the nitric oxide ligands to the iron in the heme to manipulate the activity of the protein. Why are these cofactors important? I think that one of the problems that we have with individual dysfunction, with hypertension, diabetes, atherosclerosis, etc., is that we have an oxidant insult, stress, and we make a lot of oxidant free radicals, and we deplete some of these reduced cofactors. That's why I think vitamin C, vitamin E, and some of these antioxidants are beneficial to restore the cofactors and help you make more nitric oxide. But there's some studies that suggest that, but a lot more work needs to be done with how this whole cell signaling pathway is put together. There are a variety of hormones that interact with their appropriate receptors on cell surfaces that regulate the intracellular concentrations of these cofactors, such as calcium or tetrahydrobiopterin, etc., that are required for the nitric oxide synthases to convert L-arginine in their diet to nitric oxide. The nitric oxide increases guanylate cyclase activity in the cyclo-GMP, and we can potentiate the effects of nitric oxide with phosphodiesterase inhibitors such as sildenafil and a variety of other methyls intermediates and compounds. We can come along now with a variety of tools in this pathway for potential drug leads for various diseases. We can come up with hormone agonists and antagonists. We can modify these cofactors in a variety of ways. We have compounds that will inhibit nitric oxide synthase that are in clinical trials for various diseases. We'll tell you about them shortly. There are compounds that will scavenge nitric oxide. Not only will you scavenge it with hemoglobin and myoglobin, but there are some small thiols that are in clinical trials that will suck up the NO and prevent its effect when you're making too much of it. And we also have other compounds that will activate guanylate cyclase or potentiate the effects of nitric oxide. So we've got a lot of tools to think about how to use this now for drug discovery and development. Well, one of the problems is, this is the summary of what I just showed you in the upper left-hand part here. Not only does nitric oxide activate guanylate cyclase and make cyclo-GMP, but it also does other things, obviously. It's oxidized to form nitrite and nitrate. The nitrite in your saliva, when you swallow it into an acidic stomach and an acidic environment, you regenerate nitric oxide. So your salivary glands are probably talking to your stomach. It's an interesting concept. It forms complexes with other transition metals, preferably iron, but other transition metals as well. It'll form derivatives with thiol groups and proteins. There are about 100 proteins that form nitrosophiles with nitrisation reactions. Some of these are very important. Some of these enzymes participate in what we call cell death or apoptosis. Some of them are transcription factors that regulate genes. So there are other ways to signal the nitric oxide besides cyclo-GMP. A very important pathway, I think, is this one, where nitric oxide interacts with superoxide anion to form a very reactive species called peroxynitrite. Whenever you have an inflammatory event, you induce NOS2 formation to make a lot of NO, but you're also inducing other pathways to make a lot of superoxide anion. And therefore, whenever you have NO in the environment of superoxide, it goes this way to peroxynitrite instead of activating the cyclase. So inflammation, atherosclerosis, sucks up the NO and pushes it down the peroxynitrite pathway instead of over the G-cyclase to down the blood vessel. That's what's happening. Why is the peroxynitrite important? Well, and this reaction is diffusion limited almost. It's very, very fast. it'll form complexes with proteins by forming a nitration reaction on the tyrosine residue of the protein. And we've been very busy the last few years trying to identify some of these proteins. And some of them are going to be very, very important. Some of them are important enzymes that participate in cell signaling pathways. Some of them play a role in metabolism of ketone bodies and diabetes, etc. I think they're going to be markers of inflammatory disease. They're going to participate in some of these inflammatory processes. And we wondered, is this reaction reversible? If it is, wouldn't it be nice to have drugs that push this reaction back this way so it would go down this pathway? The other approaches to arthritis treatment, say, or inflammatory bowel disease, or whatever. Let me show you some of the diseases that are being addressed with this nitric oxide biochemistry and biology. Many major multinational pharmaceutical companies have programs related to nitric oxide one way or another. They're making new nitric oxide donors, better nitroglycerins, if you will. Others are looking at delivery systems. Some are looking at nitric oxide synthase inhibitors. But besides all the activity with multinationals, which is often difficult to know about because not much is publicized until they're in the clinic usually, there are probably more than 20 or 25 biotech companies around the world now that have developed because of nitric oxide biology to find and to get a drug approved for one indication or another. These are some of the things that I think are going to be looked at. We know that nitric oxide is a neurotransmitter. We also know from work of other laboratories, Eric Kandel in New York, who got the Nobel Prize just years ago, that it participates in that. If you knock out the NOS1 and NOS3 in mice, they don't do well in the water maze test. They can't remember where the float is when they swim across the tub. So they've got a memory defect. What it is, we're not, how it's participating, we're not sure. It's obvious that nitric oxide is participating in the formation of fluid in the anterior chamber and the posterior chamber of the vitreous, and also the absorption of fluid. And it probably plays a role in the retinal nerve degeneration with patients with elevated intraocular pressures with glaucoma. So there are some groups trying to pursue approaches with nitric oxide biology to treat glaucoma. Certainly its effects as a vasodilator to control blood flow. We've talked a lot about that. A very exciting thing is developed. Premature babies and many children with congenital heart defects are called blue babies. And they're blue babies because they shunt the blood right to left. What do we mean by that? Well, the blood that comes back to the right side of their heart doesn't get oxygenated in the lung, but instead goes through a hole in the atria, a septal defect, out the left side of the heart, and through the patent ductus. As it's coming out, it's going through a patent ductus, or it doesn't go through the hole, it can just go up the pulmonary artery through the patent ductus in the aorta. Nevertheless, the blood goes from the right side to the left side without going through the lung because of pulmonary hypertension. Premature babies don't have adequate lung development. They have deficiencies in surfactant, they have pulmonary hypertension. It turns out that if they inhale very low concentrations of nitric oxide, very, very low concentrations, with their oxygen, their blood vessels in their lungs dilate, they stop shunting blood right to left, they oxygenate, and they really improve markedly. This is also true with patients with some of the congenital heart defects. So that's been very exciting. And most hospitals in the U.S. and many in Europe are now giving low concentrations of nasal oxygen and nitric oxide to premature babies. I haven't seen much use yet in Asia. I suspect that'll pick up this next year or two. It doesn't seem to be as effective in adults with pulmonary hypertension. And that may be because a lot of adults have end-stage disease and not much of a reversible element. We talked about penile erection. There will be a number of drugs on the market in the U.S. and Europe within months that will compete with Viagra. There are a lot of drugs available now from a variety of companies. And I know that there are a couple in the U.S. being reviewed right now by the FDA. It participates in angiogenesis and atherosclerosis. I told you about that. There's evidence in the plaque of an atherosclerotic plaque that you need a lot of nitric oxide. Why do we know that? Because there's a lot of nitrotyrosine-containing protein in the plaque. It's an inflammatory event. The nitric oxide is important for endothelial cells to grow, and it suppresses the growth of smooth muscle cells. So it also plays a role in root remodeling of blood vessels, and that probably is going to be important in cancer. Tumors, as they grow, have to grow blood vessels to be furnished with nutrients. If you inhibit the growth of blood vessels with these angiostatin inhibitors or perhaps other compounds related to nitric oxide, you may be able to shrink the tumor. It's probably also going to play a role in retinopathy, diabetic retinopathy, and perhaps wound healing. I've seen some very interesting data in the last few months of topical immunodonors on burns and wound healing, and I think that'll look promising as well. This is my last slide. There's evidence in transplantation of tissue that there's an inflammatory component of that when there's a mismatch with histocompatibility or the matching of the tissue to the recipient. This results in the release of various cytokines that turn on NOS2, that make a lot of NO, it becomes toxic, and it may participate in some of the rejection of that transplant. I talked about platelet aggregation, a very interesting story that I heard from a cardiovascular surgeon in Germany a few months ago. He is infusing low concentrations of nitric oxide in the heart-lung machine as he does open chest surgery for his patients. And he finds the nitric oxide inhibits the aggregation of the platelets. The platelets have better survival as they go through the machine. They're not destroyed. And therefore, the patients have less bleeding in the chest and improved recovery time. Very simple maneuver. It will have perhaps a big impact on patient care with open heart surgery. Stem cells, it's very early, and I think it's got a lot of promise. Nitric oxide, we think, is going to regulate the proliferation and differentiation of stem cells. And I think that's going to be a big area in the future of medicine for tissue regeneration, replacement, and gene therapy. I think I'm going to stop. I think there's some time for questions. Thank you very much.
So, may I invite Dr. Talat Somabut and Dr. Anek Aripak to be the moderators of the questions and answer session. In the meantime, we welcome all questions. Please write your questions in the paper, and we have our people collecting the questions, and all the questions will be transferred to the podium up there, and both of our two experts will ask the questions.
Last speaker. Shall we project on the screen, or shall I ask the question from the paper? Often associated with male sexual dysfunction such as ED. Does nitric oxide have a role in this problem, and what is the possible mechanism of the people? Answer this.
I'm not aware. With 50,000 papers in the field, I don't read but a fraction of them. It's impossible. I've got lots of other things to do with it. I don't know off the top of my head of studies with anxiety and depression. With regard to the end effect of the nitroglicergic nerves on the blood vessels in the corpus cavernosum, I think certainly there's going to be a role for nitric oxide and cyclogene P. And whether these patients are releasing less nitric oxide or some other factor, I don't know. I'm not sure what's happening. I mean, nitric oxide does lots and lots of things, but it doesn't do everything, I guess. I hope it doesn't do everything. But there are areas that we need to look at even further. I'm not aware of anxiety and depression.
Shall I pass for the next question?
And it is really, what do you think about might be the next stage of development in this field? That's the first question.
There's an awful lot that we still don't know. It would be nice to have very selective inhibitors of each of the nitric oxide synthases. If we had selective inhibitors of each of those, specific inhibitors for each, without crossing over and affecting another isoform, it would really help sort out a lot of the biology, and it would offer a lot of advantages for drug development. There are diseases where you want to suppress only NOS2. There are, for example, inflammatory disease or septic shock. There are other diseases where you want to suppress NOS1 primarily in the brain. If you inhibit NOS1 and decrease nitric oxide in the brain, you can decrease the size of an infarct and the damage to the brain with a stroke. You don't want to inhibit NOS3 because you compromise the blood flow. That's one area. We need to understand more about guanylate cyclase and have other activators and inhibitors. We have some activators that are not specific. They do some other things. We have some inhibitors that are not specific. If we had more selective activators and inhibitors, we probably could bypass the need for nitric oxide in some cases, say in erectile dysfunction or in very severe atherosclerosis, and be able to treat it better and not be dependent so much on making the endogenous nitric oxide. We need to understand the role of all of these other phosphodiesterases. It's a very complicated family. There are 9 or 10 gene products and isoforms in phosphodiesterase. Some hydrolyze cyclic A, some cyclic G. And if we had, again, selective tools, we could sort this out of the biology and really do some more important clinical studies. For example, we're still not sure what cyclic GMP is doing in the heart. I mean, here it's been now, my goodness, almost 40 years since the discovery of cyclic GMP. And we're not really sure what it does to myocardial cells in the heart. Does it alter conduction? Does it alter contractility? Does it change calcium? Does it change cyclic GMP, AMP? A lot has to be done. And I guess it's the absence of pharmacologic tools that are the most important. I think also in the future, we are going to begin to think a lot more about using the genes for these proteins, not only in the laboratory to make transgenic animals, to make knockout animals, we're going to start thinking about gene therapy in patients. And I think that that's on the horizon. Within 3, 5, 7 years, we're going to start treating patients with genes with these proteins. It's coming.
So the next question is, how can a person join particular research if he or she is interested in that field? How can a person join a particular research if he or she is interested in that field?
Well, I guess it depends whether it's a senior person who has the staff and trainees and resources to change direction, or is it a young person wanting to work in that field. If it's a young person, I would say the best way is to go to a laboratory with a lot of experience in that area and train for several years. As a senior person, if you have resources, you can do it. It just takes a while.
But the end of the question is, who funds the research?
Well, it depends on where in the world you're doing research. In the United States, almost half of our research budget in the United States comes from the government. NIH grants, NSF, but NIH predominantly. We probably, in the United States, are spending 60 to 70 billion US dollars per year on medical research. About half of that from the NIH, the other half from industry, private companies doing drug-related research. Most of that is on really transitional research, more clinical development, if you will. I know that the budgets in other countries are not of that magnitude, but I was telling someone at lunch that I think it's very important that countries, once they have improved their infrastructure, the sewers, the waters, the roads, the bridges, begin to make investments in medical research, because it's very important to quality of life. And if you don't do it, you're going to be paying a premium for healthcare by buying the technology from other countries. And you will not have intellectual property of your own to do that. And that means that there's going to be a higher price for it. So I think we've learned that one of the best investments you can make really is research to enhance the quality of life and improve diseases.
Thank you. Nick?
Well, a short question, because we have so many questions here. The question is this: is there any cell surface receptor for nitric oxide?
The first receptor is guanylyl cyclase, the cyan lysoform. Guanylyl cyclase is a receptor. It binds nitric oxide. That's the definition of a receptor. We know that it also binds to other proteins now, but that was the first one. And it'll form complexes with some heme proteins, with iron sulfide centers, such as aconitase, and with thiol groups and other proteins in nitrisation reactions. So the list is growing. There are lots and lots of proteins, but the first was the guanylyl cyclase.
One more question.
One more, okay. The question is, you want to know if nitric oxide effect on vasodilatation mediated through potassium channel.
The nitric oxide increases cyclic GMP. The cyclic GMP can do a variety of things. It can regulate cytosolic calcium phosphorylation of myosin. It can also regulate phosphodiesterase activity, and it can also regulate channels. probably calcium channels, maybe potassium channels. So it has multiple effects. And the precise mechanism to cause relaxation, we're still not sure, because it may be doing multiple things. But the outcome is really, ultimately, the dephosphorylation of myosin leaching, probably by lowering calcium, but it may do that several ways. We also know that cyclo-GMP can decrease the conversion of phosphoinositides to inositol 3-phosphate. Inositol 3-phosphate has a receptor in the sarcoplasmic reticulum to mobilize calcium in and out of from cytosol to the SR. And that reaction, that pathway is inhibited as you lower IP3. So it can do it several different ways, including some gated channels.
Well, due to time limit, this will have to be last question. What inspired you to your discovery? What inspired you to your discovery? Very good.
I love research. I think research is exciting. You don't find answers every day, but when you find an answer to an important question, there's nothing like it. I tell people that when you design an experiment from your experience, your intuition, your previous work, and the experiment works, and it happens to be a very important experiment, there's nothing like it. I have never taken heroin, but I suspect it's like taking a big slug of heroin. It's exciting. You see that data coming out of the machine and it works, wow. And then your mind clicks and say, this is the next experiment, is to prove how important it is. I think it's the love of research and science and being inquisitive to get answers to questions.
Well, thank you very much. Due to the time limit, I think we have to stop answering questions right now. But however, we will give this question to the speaker and ask him to answer all these questions. And somehow we try to return all these answers to all those who have asked these questions. Well, thank you very much.